The overall goal of this method is to use multiple reaction monitoring mass spectrometry to identify and quantify the species present in raw meat mixtures for use as a tool for food fraud detection.
This method can help answer key questions in the field of food integrity, such as confirming the species present in products like sausages or burgers. Our approach is based on mass spectrometry of the protein component. The main advantage of this technique is that it can give a rapid and quantitative outcome.
We first had the idea for this method when we noticed that proteins known by the same name in different species are actually different enough to give species identification, but similar enough to allow quantitation.
Thermally denature purified reference myoglobins by heating the sample in a hot block at 95 degrees Celsius for 30 minutes. Cool the sample for approximately 15 minutes until it reaches room temperature. Then add 30 milligrams of urea to enhance the digestion and mix. Add a trypsin solution to the sample in a 1:30 ratio of enzyme to substrate by weight. Then mix by gentle vortexing and allow it to proteolyze overnight at 37 degrees Celsius. Next, desalt the post-proteolysis sample by first diluting the sample 1:2 volume to volume with water. Activate a polymeric reversed phase cartridge filled with 30 milligrams of reversed phase material by adding 1 milliliter of methanol. Then equilibrate the cartridge.